All right, welcome back everybody to the deep dive. Last year's Nobel Prize in chemistry, you remember that, right? All the excitement about AI finally cracking protein folding. It felt like, wow, we finally figured out this fundamental secret of life. But what if I told you that was just the first step? Today, we're going deep on what's next. It's pretty mind-blowing. We're talking about learning to engineer biology itself.
Yeah, you hit the nail on the head there. Solving the monomer folding problem. Huge achievement, no doubt. But to really grasp this next wave, you got to see it as a stepping stone. The real frontier now is making biology programmable, designing brand new molecules from scratch, molecules that do exactly what we want them to do. Think of it less like reading the book of life and more like writing whole new chapters, custom made for specific purposes.
And that's exactly where our deep dive takes us today. The incredible work being done at vant.ai. They're not just predicting how things look. They're building the tools to design completely new molecules in a way that's almost like programming biology. And here's the key. They're weaving in real experimental data to make sure it all works in the real world. So today's mission, unpack their approach, especially their NEO-1 foundation model and their NeoLink data platform. We want to understand how they're pushing the limits of molecular design, especially those really innovative academic breakthroughs that are making this possible.
Absolutely. Right at the heart of their innovation is NEO-1. They call it the world's first truly general-purpose atomistic foundation model. And what's remarkable here is that it brings together two things that used to be separate, super-specialized fields. The ability to accurately predict the 3D structure of biological molecules and the ability to design totally new ones from scratch down to the level of individual atoms and how they interact.
Okay, so let me get this straight. NEO-1 isn't just figuring out how an existing protein folds up. It can also dream up entirely new molecules, things that don't even exist in nature, right? And it does this with atomic level precision.
Precisely. This merging of capabilities, it's a significant leap forward intellectually. It allows scientists like us, and ultimately this benefits everyone through new potential therapies, it allows us to give the model incredibly specific instructions about structure, not just for single molecules, but for how they interact with each other. Imagine saying to an I need a molecule that binds this very specific spot on this protein and makes these exact atomic contacts. And then it actually comes up with workable solutions.
That sounds incredibly powerful. Like you could tackle really complex biological problems that are just impossible right now. And you mentioned Neolink, right? How does that fit into all of this?
Right, so NeoLink is their proprietary platform based on cross-linking mass spectrometry, or XLMS for short. Think of it as a way to generate a fundamentally different type of structural data. It gives you these sparse data points showing which parts of molecules are close together in a complex. It doesn't give you the whole 3D structure by itself, but it's super valuable for guiding and validating what a model like NEO-1 predicts.
Okay, so we've got this advanced AI, NEO-1, that can both understand and design molecules at the atomic level. And we've got NeoLink providing it with real-world structural clues, like breadcrumbs leading to the complete picture. Now, let's dive into this shift beyond protein folding. You were talking about neoproteins. What's the main idea there?
What's really innovative here from an academic standpoint, and this will eventually have an impact on everyone, is vant.ai's core vision, making protein interactions programmable. They're not always trying to build new proteins from scratch for every therapeutic need. Instead, they're figuring out how to cleverly use the huge variety of proteins we already have in our cells. These naturally occurring proteins, when we redirect or modulate their function, that's what they call neoproteins.
Okay, so how does this reprogramming actually happen? What's the mechanism?
They're pioneering something called proximity modulation, or PROMOD. The basic idea is to use small molecules, peptides, or even these larger macrocycles as guides. These guides direct the neoproteins to specific targets in the cell. It's like hijacking their normal biological activities to achieve a specific goal, like fighting a disease. You're basically rewriting existing cellular machinery to do something new and beneficial.
That's clever. It seems more efficient than trying to create something completely new every time. But you said traditional drug discovery methods run into problems with PROMODs. Why is that?
Right, so traditional structure-based drug design, it relies on having a stable, well-defined 3D structure of the target protein. But with PROMODs, the complex that matters the neoprotein bound to its target, with the help of the modulating molecule, that complex is often only stable and measurable when the modulating molecule is actually there. So it's incredibly hard to figure out the structure of that complex using traditional methods before you even start designing the molecule.
So it's a catch-22, right? You need the structure to design the molecule, but the structure only forms when you have the molecule.
Exactly. And that's where NEO-1's unified capabilities come in. It's a huge academic breakthrough. vant.ai realized that to design these neoproteins and the molecules that guide them, you need a model that can simultaneously predict how the whole complex will fold and design the molecule that brings them together. Previous models couldn't do that. They treated those as separate steps.
This sounds like a fundamental change in how we think about molecular design. So on a technical level, how does NEO-1 pull off this feat of decoding and designing the structure of life, as they put it? What's the core novelty?
The key advancement, and this is fascinating from a machine learning perspective, lies in how NEO-1 represents molecules during the design process. Traditional methods treated structure prediction and molecule design as totally separate computational problems. NEO-1 changes that. It combines state-of-the-art structure prediction and all-atom molecular generation into a single unified neural network. They did this by moving the diffusion process, which is a core technique in generative AI, where you refine a noisy starting point. They moved it from the normal 3D space of atomic coordinates to something called a latent space.
Latent space. Can you explain that a little more?
Think of latent space as this compressed, abstract representation of all possible molecules and their structures. By doing the computationally heavy lifting in this abstract space, NEO-1 can reason over a much smoother, more continuous landscape. It elegantly captures the relationships between a molecule sequence, like the amino acids in a protein, and its complex 3D structure. This lets it generate entirely new molecules, proteins, peptides, small molecules with atomic precision, while also predicting their structures very accurately. Like the model has learned the fundamental rules of both the building blocks of life and their final architectural arrangement, all at the same time.
So it's not just sticking a molecule generator onto an existing structure prediction model. It's a much deeper, more fundamental integration.
Exactly. This shift in architecture leads to a new level of generality and programmability. NEO-1 can handle a whole bunch of tasks that used to require separate, highly specialized models. Think about co-folding multiple proteins, predicting how a drug-like molecule will fit into a binding pocket, designing a protein sequence that folds into a specific ship, we call that inverse folding, even creating totally new proteins or small molecules with specific properties. All these different tasks now handled by one model.
Wow, that's incredible versatility, like a Swiss Army knife for molecular biology.
Precisely. And this all stems from its ability to learn what they call a learned, unified, latent representation of different biomolecules. It learns to compress the essential information about small molecules, lipids, proteins, even DNA and RNA into this shared abstract space. And crucially, it can decode this representation back into a complete, atomistically detailed molecule with all its atoms in their exact 3D positions.
And this is where the programmability comes in.
Exactly. Because NEO-1 learns this underlying representation, you can give it incredibly specific instructions. You could give it a protein's amino acid sequence and ask it to predict its folded structure. You could give it a partial 3D structure and ask it to fill in the missing parts. Or, and this is important for the ProMod approach we talked about, you could give it the sequences of two proteins that interact and ask it to design a small molecule that binds to both, effectively gluing them together and folding the whole complex. You can even add extra information like what type of molecule you want, the binding site on a protein, or special distances between atoms to guide the design.
So it's taking specific instructions and using its knowledge of molecular principles to generate potential solutions.
Exactly. And then there's steerable molecular generation, which is even more powerful. Unlike some models that build molecules step by step, atom by atom, NEO-1 works more holistically. It generates its predictions in a course-defined way. This allows you to guide the process towards specific goals. Let's say you want to make a drug candidate more rigid, which often means it'll work better as a medicine. You can apply constraints across the whole molecule, even for properties that are hard to compute. The model learns to optimize for these complex properties without needing to be retrained for each new objective.
That level of control seems really useful for fine-tuning drug candidates. Okay, so NEO-1 is the brains of the operation. Now tell us more about Neolink and how it feeds this AI with real-world data.
Neolink is vant.ai's platform for generating structural data on a massive scale. They compare it to how shotgun sequencing revolutionized genomics. Just like shotgun sequencing breaks the genome into small pieces for sequencing, Neolink uses XLMS to introduce these chemical rulers between parts of molecules in a complex. These rulers measure the distances between specific atoms or amino acids. This process feeds what we call sparse structural restraints. They tell us which parts of molecules are close together in 3D space.
So sparse means it doesn't give you a complete high-resolution structure, just some key distance measurements.
Right, it's like getting snapshots of a scene instead of a full panorama. The power of NEO-link is that it can do these measurements really fast and cheaply, generating tons of information about how molecules interact in a cell. Vant.ai says their NEO-link data already covers a huge chunk of the human proteome, more than what's in the public protein databank.
Wow, that's a lot of data. But it's sparse, like you said. How does NEO-1 use these fragments to build a full picture?
This is where NEO-1 and NEO-link work together beautifully. NEO-1 is designed to take those restraints from NEO-link and assemble them into high-resolution 3D structures. It's like using bioinformatics tools to piece together the short reads from shotgun sequencing into a complete genome sequence.
So NeoLink generates the raw experimental data, and NEO-1 makes sense of it to build a detailed structural understanding.
Exactly. And this creates a feedback loop. As NEO-1 gets better at understanding the NEO-link data, its predictions become more accurate. And those accurate models inform the design of future NEO-link experiments, which then generate even better data. It's a continuous learning process. vant.ai calls NEO-link a black box platform because it's highly automated and made for AI interpretation.
That's a clever way to connect experiments and computational modeling. So how well does NEO-1 actually perform? You mentioned comparisons to a model called Boltz-1, right?
Yes, vant.ai compared NEO-1 to Boltz-1, which is an open-source version of DeepMind's AlphaFold 3, a top protein structure prediction model. Overall, NEO-1 does really well, comparable to Boltz-1, and its strengths reflect the data it was trained on. It's particularly good at predicting the structures of protein-binding pockets, protein-protein complexes, and especially those ProMod-induced complexes we talked about. Interestingly, it's slightly less accurate for single proteins, probably because it's focused on those more complex biologically relevant structures.
So it's designed for the tough cases, the ones that really matter for drug discovery.
Precisely. And it shines in those really challenging situations, like ternary complexes where three or more molecules interact, antibody-antigen interactions, and protein-peptide complexes. They specifically highlighted its performance on PROTACs. PROTACs are a type of promod that are really hard to predict because they involve interactions that don't have a strong evolutionary history. NEO-1 beat Boltz-1 on a recent PROTAC dataset, and its accuracy got even better when it used even a little bit of NEO-link data.
That's a big step forward. And you mentioned NEO-1 predicting unknown complexes, right? What does that mean?
This is where it gets really exciting. NEO-1 was able to predict the structures of protein complexes that had never been experimentally characterized before. One example is the interaction between ATP-AF2 and ATP-5F1A, which is important for making ATP synthase, the energy factory of the cell. We didn't know the structure of this complex in humans, but NEO-1, using just one crosslink from NEO-link, predicted it. And its prediction matched what we already knew from biochemistry. Another model actually violated that experimental crosslink.
So it's not just refining our understanding of known biology, it's actually uncovering new biological insights.
Absolutely. Another example is the complex between UBE3D and CPSF3. CPSF3 is involved in RNA processing, and UBE3D protects it from degradation. Again, NEO-1, using a single crosslink, predicted a structure that matched the experimental data. And it even worked for a flexible region of CPSF3, which is hard to predict with other methods.
That's pretty amazing. Can NEO-1 also use structural information from databases?
Definitely, it can take existing 3D structures of proteins or small molecules and use them as input. It essentially acts like a very accurate docking method, predicting how those molecules will interact. They found that giving NEO-1 these known structures makes its predictions much more accurate.
So it can use all sorts of structural clues, whether they're sparse hints from NeoLink or full structures from databases.
Exactly. This flexibility and its ability to learn from different data sources are key to their vision of AI as a co-pilot in drug discovery. They see a future where drug designers, experimental data like NeoLink, and AI like NEO-1 all work together. As more experimental data comes in, NEO-1 can refine its predictions and designs, speeding up the process of finding new drugs.
You mentioned some case studies where this co-pilot idea is really demonstrated. Can you tell us more about those?
Sure, one case study was about discovering molecular glues and traditional small molecule inhibitors. They showed how NEO-1 could generate potential inhibitors for a protein just from its amino acid sequence. It could even rediscover features of known inhibitors while suggesting completely new chemical structures. For molecular glues, they showed that NEO-1, given the sequences of two proteins, could design small molecules to hold them together, even replicating features seen in known glue complexes. They also showed how NEO-1 could be guided to optimize for drug-like properties like QED, which measures molecular complexity. And finally, they showed how, given a chemical scaffold that binds to a target, NEO-1 could expand on existing lead molecules, even considering the protein's structure.
That's really impressive. What about designing larger molecules, like antibodies?
They had another case study on antibody discovery. They used the SARS-CoV-2 spike protein as an example and showed how NEO-1 could be used to design new antibodies against it. The model could predict the structures of antibody fragments bound to the spike protein, generate sequences for the important CDR regions of the antibody, incorporate constraints to target specific parts of the spike protein, include sequence motifs associated with strong binding, and even use existing antibody structures to improve its designs. The key takeaway was that NEO-1 could combine information from different sources, sequences, structures, experimental data to design antibodies.
It sounds like NEO-1 can really learn from and build upon existing knowledge.
Absolutely. They do acknowledge that AI models like NEO-1 still have limitations, but they stress that the expertise of drug designers and the use of high-quality experimental data are crucial to overcome those limitations. Look at ahead. vant.ai wants to understand how molecules behave in living cells, not just their static structures. They want to use NeoLink to study how molecules interact in real time and how those interactions affect drug effectiveness and side effects. They're also going to keep releasing new NeoLink datasets to the scientific community.
So while this is a big step forward, it's still early days in this new era of programmable biology.
Exactly. NEO-1 and NeoLink together are a significant step towards a future where we can engineer biological systems with incredible precision and understanding.
So the big breakthroughs here are NEO-1's ability to both decode and design molecules, its use of latent space, its programmability, and its integration with NeoLink's experimental data.
That's right, and these advancements are especially important for targeting difficult proteins and for developing new therapeutic approaches like ProMod, which have huge potential for treating diseases.
It really makes you think, doesn't it? Imagine designing molecules with this level of control. How could that change medicine and the life sciences? Could we engineer solutions to diseases in ways we can't even imagine yet? It feels like this new era of programmable biology is opening up a whole new world of possibilities.
Absolutely. It's an exciting time for scientific discovery and innovation.
If you're interested in learning more, we've included links to vant.ai's website and their research papers in the show notes. Thanks for joining us on the deep dive.
Hey there. Want to share your thoughts on this?
They changed the architecture diffusion model to from the convolutional feature to the latent space feature. What does that mean? Can you explain it?
That's a great question. Okay, let's unpack this. You're asking about how NEO-1 changed its architecture, specifically the diffusion model. You mentioned it moved from a combination of features directly to the data space feature.
What's fascinating here is the shift from working directly with the complex data of atomic coordinates to this more abstract latent space that we touched on earlier. Think of it this way. Instead of trying to build a molecule atom by atom in 3D space, which is incredibly complicated with all the possible arrangements and bond angles, NEO-1 learns a simplified, almost like a compressed code for representing molecules. That's the latent space.
Exactly. So the combination of features you're referring to could be things like the sequence of amino acids or some initial structural information. Previously, a model might have tried to directly translate those features into the final 3D coordinates, the data space.
But you're like trying to describe a whole symphony by just listing the instruments. You miss the harmony in the overall structure. By moving the diffusion process to the latent space, NEO-1 learns the underlying rules and relationships that govern how sequences and structures are connected in this compressed representation.
Here's where it gets really interesting. The diffusion model then operates in this smoother, more manageable latent space. It starts with a noisy representation and gradually refines it into a coherent molecular code. Only at the very end does it decode this latent representation back into the full all-atom structure in the data space.
So what does this all mean? By doing the heavy lifting in the latent space, the model can reason over a much broader range of possibilities and capture more subtle relationships between sequence and structure. This is key to its ability to not only predict structures, but also to design entirely new molecules with specific properties, because it's not constrained by the complexities of directly manipulating atomic coordinates from the start.
It's like learning the grammar of molecular structure before trying to write a whole novel. It makes the whole process much more efficient and allows for that simultaneous understanding of both building blocks and final shapes we were discussing.
Precisely. And this architectural choice is what allows NEO-1 to unify those traditionally separate tasks of prediction and generation within a single model. It's all about reasoning in that learned, simplified representation.
Okay, now that we've hopefully clarified that shift to the latent space, let's circle back to how this enables that broad generality we mentioned.
Right. Because NEO-1 has learned this underlying representation, it's not limited to just one type of molecule or one specific task. That unified latent space acts as a common language for proteins, small molecules, and everything in between.
So when we talked about it handling tasks like co-folding, docking, and de novo design, all within the same model, that's a direct result of this architectural shift to the latent space diffusion.
Exactly. It's this ability to understand and generate molecules in this abstract space that allows for such versatility. Think back to our Swiss Army knives analogy that versatility comes from having a set of fundamental tools that can be applied to different problems. The latent space is that set of fundamental molecular understanding tools for NEO-1.
And as we mentioned, this also allows for that powerful programmability. By manipulating the latent representation, we can guide the model to generate molecules with specific features or for specific purposes, like binding to a particular site on a protein.
Yes, and that steerable molecular generation we discussed, where you can apply rewards across the entire molecular structure to optimize for properties, that's also made possible by the way NEO-1 operates in this latent space. It has a more holistic view of the molecule throughout the generation process.
So this move to latent space isn't just a technical detail, it's a fundamental change in how the AI understands and creates molecules, and it underpins all those impressive capabilities we've been discussing.
Absolutely. It's a core innovation that allows NEO-1 to push the boundaries of what's possible in computational biology and drug discovery.
Well, that was a fantastic question, and I hope that helps clarify what that architectural shift means. Now, where were we? We were just about to dive deeper into some more examples of how NEO-1 is being used in real-world drug discovery scenarios.
Exactly. We wanted to highlight how this ability to both predict and design, fueled by NEOLink data, is really changing the game in the lab.
So if you're still with us, we're going to jump into some specific case studies that really showcase the power of NEO-1 as an AI co-pilot in the journey to develop new medicines.
And these examples really bring to life the potential we were just discussing, the ability to engineer biological solutions in ways we previously couldn't imagine.
All right, let's dive back in.
Sounds good.
Thanks again for that insightful question. It's great to know you're following along so closely.
Indeed. Your engagement really helps us to make sure we're explaining things clearly.
We've really enjoyed this deep dive into NEO-1 and NEOLink with you today.
And we hope it's given you a glimpse into the exciting future of programmable biology.
Until next time on The Deep Dive.
Take care.